Good morning. Thank you for joining us today. We have two outstanding experts in the field of research specific to clinical trials and the conversations that we are hearing about vaccines in our country right now. I hope that today's conversation with Dr. James Powell and Dr. Carl Victenbaum will give everyone an opportunity to better understand the new drugs, the new vaccines, how those processes develop, understand and overcome, some of the historic reasons why we have mistrust as a Black people in this country, and consider what and how and why it is important for us to participate in clinical trials and what that really means for us relative to not just the COVID vaccine, but in medications that are developed to treat many of the diseases that we are all grappling with. And then finally, how patients can navigate and understand why and how the vaccine's created, and the role that you play, and how you come to the decisions that you make relative to participating in vaccine trials as well as taking the vaccine. So I'm honored today to introduce our first panelist. And before I do that, though, I would like to share with you Dr. Lou Edgy, who was to join us today, is actually giving testimony to the American Medical Association on policy. And so she will not be able to join us today, but I know that Dr. James Powell and Dr. Carl Victenbaum will give us great information and help us build a conversation that we don't see this as a beginning and end conversation, but a starting point to making sure that we understand what we need to understand around the science. So Dr. James Powell is someone I've known for a long time, and he has been a long time advocate and scientist in addressing why and how the need for Black people and brown people to participate in clinical trials. Is passionate and is a noted expert across this country. Dr. Powell was born in Birmingham, Alabama. He graduated from Virginia Union University and Cornell University Medical College. He was trained in clinical pharmacology at Cornell and taught there a while before joining Procter and Gamble in pharmaceutical research. He is the former member of the Schulman IRB, which is the Health and Human Services Secretary's Advisory Committee on Human Subject Protection. He is the principal investigator of the National Medical Association's Project Impact, and Project Impact is a project that is focused solely on why we must be a part of clinical trials. He is the chief medical officer and co-founder of NoRx. Please join me in welcoming Dr. James Powell.
Thank you. Thank you very much, Renee, for that very kind introduction. I'm going to see if I can get the screen going with my slides here. And go to here. Okay, let me see if I can go. Okay, this is the end. Okay, and let's see. All right, there we go. Thank you. And thank you all for being at this discussion. I always look forward to doing this kind of discussion. As Renee said, that I am very passionate about the issue. And as I shared with one group earlier, that I've been known from time to time to sit in the barber chair, and we have a discussion in the barber shop and about clinical research participation. And one time I did it, and the barber kept cutting while I was talking. And the next time when I got up, I didn't have very much hair, but at least everybody was reasonably well informed. But I'm going to talk to you a little bit about understanding the development of medicines. And later, when I complete this, Dr. Victenbaum is going to talk a little bit more about the vaccine clinical trials. But I want to start singing from the same hymnal with respect to how drugs are developed. I really got involved in this because very early on in my life, growing up in Birmingham, I was very concerned that the people such as myself, and primarily my brother, who was often involved in the medical care system because he was born with some defects, birth defects, and I got to be concerned that people like me weren't represented in the medical profession, in the medical understanding of medical disease. So I focused on the issues of drugs and drugs development. And what I'm going to share with you just a little bit, go quickly about this slide, is talks about the drug development process. And we start by when there's an idea or data that suggests there's a way to address a particular disease, then the work begins in the laboratory to try to understand how chemistry can impact biological systems to produce a beneficial effect. And it starts off with doing some studies in animals to determine the drug's effects, safety effects on two or three species of animals prior to introducing those new medicines into human beings. That happens when you file that information from animals to the FDA and gets what's called an investigational new drug that permits you to evaluate a drug that's not approved in people in the United States. When you have that approval to do that, move forward, you do what's called phase 1 testing. And that's generally safety and tolerability and what's called pharmacokinetics. And that really addresses what the body does to the drug when the drug is introduced. And we'll talk a little bit more about that in a few minutes. But once you get safety and tolerability for that new chemical, it's not a drug yet, until you can introduce it into patients. For instance, if you have a diabetes patient, you go to phase 1 may involve 20 to 100 people to show its safety and tolerability. But then you move to phase 2, where you have patients to be able to try to understand whether or not the product has a desirable effect in human. For instance, in the diabetes trial, you may take patients and look and see whether or not that chemical, when introduced to people, will in fact lower blood sugar. Once you've done that and shown that it actually has an effect in patients, you will begin to do other studies to look for the appropriate dose in the patient population. And those are both part of phase 2 trials. Once you have both the right dose that delivers an effect without minimizing the potential for risk, then at that time, those developers will have the opportunity to share that information with the FDA and decide what's going to be done in what's called phase 3. And phase 3 is the phase when we do many more patients to be able to confirm their safety and effectiveness in human beings with the disease. And you do that, usually it's at least a couple of trials. Usually those are randomized, often double-blind, placebo-controlled clinical trials, meaning that neither the investigator or the patient know what treatment is given. And placebo, of course, is comparing the active, what you expect to be the active ingredient to something that is not active, such as a sugar pill. Once you have that information, not only the effectiveness in those trials, you proceed, you also collect the information with respect to any kind of adverse events, safety issues. And it's important there because all the safety issues that you've observed in all that clinical trials is compiled, and that becomes what in the label with respect to the risk associated with those medications. And so you file that with FDA to have what's called a new drug application for a new drug, or for a biologic such as a vaccine or other kind of antibody. It would be filed as a biologic licensing application to the FDA. And if it looks good, then the FDA will allow that to be approved. Now it's not, as I said, not just proven safety and efficacy, but you want to do also long-term safety with a drug or active ingredient to be able to determine, okay, what's the potential of those kinds of effects to occur in a marketplace, because you recognize, as you see there, probably in the clinical trials, you probably expose a drug to maybe 5 to 10,000, but in vaccine cases, maybe as many as 30 to 60,000. But recognize that when the product is approved, it's going to be exposed to perhaps millions of patients. So you do the best to understand the safety and effectiveness preclinically. Now, one of the things, the reason it's so important that we have diversity in these clinical trials is we need to know whether or not the product works for the particular patient that's going to use it, the diversity of patients that are going to use it. Genetics, ancestry, nutrition, environment, other habits and practices, all those things might be associated with race and ethnicity in this country, around the world, actually. And all of those things can affect what I mentioned before, the pharmacokinetics, or what the body does to the drug in the body. How well is it absorbed? How well is it distributed to the site of action? How is it metabolized? What system metabolizes it to break it down to something that can be excreted from the body? And so all of these factors will determine how you respond to a particular drug. Now, it is important that we have participation across the potential users in the clinical trials, because we want to know whether the effectiveness is the same for all the populations that will potentially use it, and also whether or not the risk and the safety is the same for the different populations using it, and to be able to determine the degrees in which they're different, and so that the physician in dealing with the patient in front of them can be able to make the decision regarding the correct dose to use in that patient, or whether or not to use that particular product in that patient at all. The unfortunate fact is that African American and other minorities don't participate in trials as frequently as whites do. And this, since 2012, the FDA has been required to issue reports about the demographics of the population that's actually in the clinical trials that led to approval of drug. And the fortunate point is that up to 80% of the drugs annually indicate there's inadequate data to be able to prove the safety and effectiveness in non-white patients. And that is of great concern, because then that says that when we're using the product, then we may not have all the data, all the guidance give all the guidance to the physician as to how to use that drug safely and effectively in the diversity of patients that may come to them for treatment. And I put one particular example down there for a drug that was approved in 2015. What you see is that 88.7% of the patients that were used for the approval of that heart failure drug were white patients, whereas only 1.2% African Americans, and you see there for Asian and other patients. And what that says is that we really cannot confirm or do not have sufficient data to show whether or not that product is in fact safe and effective for a disease that we know occurs quite frequently in the African American community. And that is of concern. Now, I don't want anything that I say today to be misconstrued as telling people not to take their medication. When you have a physician prescribe you a medication, one that you trust for your care, then you need to take your medication. But what is important is that those physicians also be aware of this kind of data to be able to monitor you appropriately, knowing in fact that there may not be as much data for people like you supporting the use of the drug, use that particular drug. Now, quite frequently people talk about the reasons that patients don't participate in clinical trials is mistrust. And as we in the African American community know that we have a lot of reasons to mistrust the research process, and those reasons began from the very time African Americans appeared on these shores. And that, in fact, a couple of big incidences was certainly the research that was done by the individual that was known as the father of gynecology, who did surgery on slave women. I think he did, in one particular case, he did 35 surgeries on one woman, gynecologic surgeries on one woman without anesthesia. And so for a long time during slavery, people were involved in experiments and had no right to resist participation in those experiments. Now, since, of course, slavery, there have been other occasions where people have been abused. We know in Jim Crow era, people weren't always informed about the research they might have been exposed to. And people in Cincinnati, of course, know about the Cincinnati radiation experiment. But most often, I've done presentations across the country in many, many communities, and I've never been to one where someone didn't bring up the Tuskegee experiment. And that's the United States Public Health Service's study of syphilis in the Negro male at Tuskegee, Alabama. In that particular experiment, as you know, as most of us know, that 399 Black males with syphilis were allowed to remain untreated for 40 years with syphilis. And in fact, those experiments that were taking place were spoken about in medical meetings, and it wasn't until the lay press got a hold of the story that it became public knowledge, and a number of people expressed their concern that this was entirely unethical for this to be conducted, especially in the United States of America. So one of the things that resulted from Tuskegee, as well as some other experiments, I talk about the Willowbrook experiments, where mentally retarded people were injected with hepatitis, and a number of other things that led to what's called the Belmont Report. And that was a national commission that I think was appointed by, I think, President Carter, to review ethics of clinical human subject protection and research in the United States. And from that, we had three fundamental principles of research. And that is respect for persons. That means that persons make the decision for themselves whether or not to participate in clinical trials. And those people who are unable or vulnerable and unable to make those kinds of decisions must be protected from research. Beneficence, where essentially the benefits of that research must outweigh the risk, and you must minimize the risk in the conduct of that clinical research. And justice, meaning that the risk of the research is generally borne by the people who will benefit from it. Now, the way that's applied in the US today is that we have informed consent where patients give their permission to participate in research. We have protocols that describes exactly what is being done. And one of the things about informed consent, they must disclose to you all the risks and benefits and whatever choices of therapy you may have before you agree to participate in that research. And it is not a contract. One can withdraw from their informed consent at any time during the process of research. And then there's an institutional review board. These are groups that review the protocol in the clinical trials to make sure that are appropriate to be conducted. And they generally should include people from the community. That includes physicians, other scientists, laypersons, individuals that are not associated with research to give permission to allow that protocol to be actually conducted in that community. And you should know that the Tuskegee experiment did not have a protocol. Now, one of the things I often say is that, and forgive me if this offends anyone, but how can an experiment with 400 people, 400 men kill millions of people? Well, it's because, unfortunately, in our community, as I said, a lot of people point to that experiment as to why they don't participate in clinical trials. But unfortunately, from time to time, it's so, as I mentioned here, that the concerns about inappropriate research participation sometimes influence people from actually seeking medical care when they need it, and that's unfortunate. So, but what we do know, and what has been published, and what I've learned from the work that I've done with Project Impact, is that racial and ethnic minorities tend to participate at similar rates as white when they're asked in a manner and given the information in a manner that they can understand it. When they're informed properly, people tend to participate at the same rates from all different communities. So what do we need to do to overcome the barriers? I'm counting on you, as well as all the physicians in our community and other people that I talk to, that we gain and share knowledge about the differences in response. We advocate for diversity at every level of the process. And what I feel is most important is that we don't allow the anticipated mistrust to be used as an excuse for poor effort to engage the community in the discussion of research, proper research. And then we must confront the fear of mistrust with the knowledge and understanding of research value and protections. And we work to support trusted physicians as investigators in our community, and hopefully that begin to influence and own the process of research in our community. And we learn and teach others to ask the right questions. And one of the things we've done, and I'm not going to go through all this, one of these things that we've done with Project Impact, we've created what's called a brochure called You've Got the Power, that gives a lot of background information, but it also gives people questions that they should ask regarding whether or not, whenever they're asked to participate in a clinical trial. And that brochure is available to you from the National Medical Association. So I'm going to complete my part of the presentation by saying that health equity for a diverse population demands a new paradigm of provider community partnership for clinical trial diversity and inclusion as a common path to precision medicine. And what I'm talking to you in that today, and the reason I'm talking to you is that I want you to have a role in it, and you need to decide what your role will be for helping to inform our community with knowledge and understanding of clinical research. So with that, I'm going to stop my screen share and send it back to Renee.
Dr. Powell, thank you. For everyone joining us today, we will have a question and answer period following our next presenter. So our next presenter is local and is national in his notoriety and the work that he has achieved. Dr. Carl Victenbaum is a professor of clinical medicine and associate chairman for translational research in the Department of Internal Medicine at the University of Cincinnati. He graduated from the University of Missouri, Kansas City with a combined BA and MD program and completed his internal medicine pediatric residency at Bridgeport Hospital at Yale University. He completed his postdoctoral training in AIDS and infectious disease at Yale and Washington University as well. He joined the faculty of the University of Cincinnati in 1999. He is the principal investigator of the infectious disease researches that is funded by multiple grants, multiple federal grants. He is the principal investigator of the AIDS Clinical Trials Group Clinical Research Site, where he is an active clinical research lead focused on HIV infection, as well as the COVID research and translational science. He is co-chair of UC's Academic Health Center Innovation Committee, charged with leading COVID research efforts. He is joining us today as the principal investigator on the Mrna trial, and will be speaking more about that with you shortly. We are very fortunate to have Dr. Carl Victenbaum, a local expert, as Dr. James Powell as a local expert, and who knew that we had such great expertise and knowledge in our city. And so without further ado, I introduce to you Dr. Carl Victenbaum.
Thank you, Renee. It's really an honor to be here. It's always an honor to be on the same stage with Dr. Powell and with yourself as well. I'm sorry that Dr. Edgy couldn't join us. She's terrific as well too, and I appreciate having time to present. And I'm gonna share my screen so I can present my slides, and hopefully I will be sharing the right slides. Let me start at the beginning. Nope, that's not the beginning. All right, we'll make everybody dizzy here for a second while I get to the beginning of my slides. I don't know why it didn't start at the beginning. All right, so I'm going to talk a little bit about vaccines, but I want to give just a little bit of background of where we are. I think it's important to note that I do research that's sponsored not only by the federal government, National Institute of Health, but also by a lot of industry partners. That is the paradigm that we use in this country to develop new therapeutics. I'm always very careful now when I give talks to say that information is changing very fast. And you know, sometimes it's hard to know whether what I say could be completely different tomorrow. I haven't ever jumped out of a plane, but I'm not sure that I ever want to. So we'll just see. So I think it's important to reflect back on where we are. And if those of us who are students of history Remember that there was the Spanish influenza in 1918. It actually lasted as a pandemic for 3 years, resulting in an estimated almost 100 million deaths. And it did affect greatly economic activity with a GDP loss in many countries, 11% in our country. It was a tremendous problem. Now we are faced with a new scourge that has come across the earth. And this was a novel virus that was identified initially and reported in December, but it was in fact discovered in November in Wuhan. And what happens to people is they get sick. Now, it turns out that probably half the cases people are infected with this virus and have no symptoms at all, which makes it really dangerous because how do you know you have it and that you can accidentally transmit it to somebody else? That's really hard. On average, it takes about 2 to 14 days for people to get sick, and you can have anything from a cold to a terrible pneumonia to get very ill, having heart problems, heart injury, requiring ventilators, and making people sick. And it turns out that that's really only in the situation of about 20% or less of people who are affected by COVID illness itself, which is different from those who are just infected but don't have any illness at all. So where are we right now in the epidemic? And these are some pictures which are not very pleasant. If you look to the left of this screen, you'll see the United States, where there's even one little area in green, a lot of orange, yellow, and very little red, meaning we're okay. We don't have a lot of cases, but that was in September, and this is now. And almost every state in our country is red, meaning that is a lot of transmission going on. And when you break it down by the county level, you can see there's lots of counties, even within the areas that are not red, that are still having a lot of transmission. This means we now are having a running count, 54 million and counting, 1.3 million deaths and counting worldwide. In the United States, it's almost 11 million people. We will easily be 11 million people tomorrow. We are closing in on 250,000 deaths in Americans. And we have 45 cases per 100,000. And on September 12th, it was 11 cases per 100,000. And that's a huge difference in a numbers perspective. It's 4 times as many cases as we're seeing. And the numbers here in Cincinnati in our area are rising rapidly. Why is that? Well, transmission occurs between close contact. You can see this little picture. This is somebody coughing, and you can see the particles spread. But even breathing, singing, talking loudly, the virus can be spread from person to person, not just by droplets, but by aerosol transmission, which means that if you're in a smaller room with somebody, transmission is possible. Most of the time, most people are catching this from their close family or their friends, people they live with or people they work with on their job. So I wanted to switch gears after giving you a little bit of background to say, well, how are we going to attack this problem? So we know that we can mitigate the disease, but to stop the disease, we have to really shut down our communities. We have to shut down our economy. We have to close the doors. We have to stop people from gathering, because right now you can see that it's really out of control when we stop doing those measures. So the next way we think about this is, can we develop a vaccine? And this is a schematic of how we normally develop vaccines. We begin to understand what a problem is. We might do animal studies. Then we begin the initial phase of studies in humans to see if it's safe, figure out what the right dose is. and then see if it works, and then it gets approved. And this is oftentimes a 10 to 15 year timeline. But what are we doing now in 2020? Well, now we're making everything compressed to a 1 to 2 year timeline, doing the experiments quicker, doing the phase 1 quicker, the phase 2, on to phase 3, and approvals, manufacturing and distributing. Some of this you can see overlaps, which helps the time. There is risk involved in that because, for example, if the vaccine doesn't work and we're already investing this and we're starting to manufacture it, spending lots of money, we may wind up with something that doesn't work. And I'll come back to that point in a minute. So all of the steps are being followed. What's happening is we're compressing the time to make this faster. Now, why are we doing this? Well, how can we do this? Well, we've actually had hundreds of years of experience with vaccine development, back from the very first vaccine that was ever developed in 1796, Edward Jenner working on smallpox. We have biotechnology that allows us to identify new viruses within weeks. So if you remember back when I first started my career and got very interested in research, I was interested in HIV. Why was I interested in HIV? Well, because at that time in the early 1980s, it didn't even have a name, and because people were being discriminated against. And I have a history within my lifetime and my family of social justice causes. And so I was very interested in trying to become involved in an area where I felt discrimination was going on with people, and people were not being treated correctly. People who were from Haiti were being put in cages in Florida because we thought they were transmitting HIV. And so this injustice bothered me. And so at that time, it took us 2 years from the initial time that the first cases of HIV were reported in 1981 until we actually really discovered the virus, and it wasn't published till 1984. If you fast forward another decade or so, we had something called the Four Corners virus. It was a hantavirus in New Mexico, Arizona, that was killing young people with a terrible pneumonia. It took us 2 weeks from the time that that was identified to identify the virus that caused it. And similarly here, fast forward to 2020, 2019, the virus emerges in Wuhan, and investigators in China identified that within 1 month's time. We were able to sequence that virus. They shared that sequence graciously with the rest of the world. We were then able to identify immediately vaccine targets. Why? Because we have hundreds of years of vaccine experience knowing what works and how to target viruses. And we then repurposed much of our vaccine technology in animal models and other models to test, are we right? After we did the test tubes, we went right to the animal models. And then several companies began developing in phase 1 and phase 2 candidate vaccines within months, and phase 3 vaccines within months. All of this was possible by a great human effort and a redirecting of scientific expertise and resources commensurate with this great task at hand to try and solve a worldwide scourge. Now, what is the understanding and human cost of this? Well, there's 2 sides of a coin always, right? Potential for success and the concerns that people have. Well, so if we think about some of the successes, well, science can accelerate when necessary, and we have the technology. In my mind, a pandemic like this equals necessary because there's a lot of human suffering. Proper resources can be put into a question and we can accelerate results. We have examples of that in history. The Manhattan Project, putting someone on the moon. When we concentrate our resources, we can actually do good faster. safety data can be generated. The same safety data is being generated that we always generate for vaccine studies. It's just happening in a shorter time window. And the same effectiveness data. We are still studying the same number of people that we do for any other vaccine. We always study 20, 30,000 people to see if a vaccine works. And then the vaccine will really signal the end of this pandemic. And nothing else is going to end this other than a vaccine or time. And so the question we have to ask ourselves, are we willing to wait 2 to 3 years till this fully affects society and suffer the costs of millions of Americans dying, or do we want to have a vaccine and prevent that from happening? Now, the concerns on the other side, well, this seems too fast, it must be unsafe, right? It's happening so quickly. Is there sufficient time to really know the side effects? And what about people who have political or other financial agendas, right? Are they interfering in this? And that causes us to have worry and concern about, is it safe? Is it going to work? What about the existing concerns about vaccines already within the public? Some people are not trusting vaccines in general. And then there's a huge risk of investment. We are investing billions of dollars and we could wind up with nothing, right? Now, that's not going to be the case, but that is a risk. And then, of course, there are lots of myths about vaccines. For example, that when you get the flu vaccine, they're actually giving you influenza. That's a myth. We only use parts of influenza. We don't use the whole virus when you get a shot for a vaccine with influenza. Some people are thinking there's microchips that are going to be inserted in you when we give you a vaccine so you can track people. There's all sorts of interesting myths out there in society. So people have legitimate concerns, real concerns, and there's real potential for success, and we have to think about them. So we have to have a framework. And some investigators from Johns Hopkins and Texas State University, the Department of Anthropology through National Science Foundation, did some thinking about this. And what they came up with were some general guidelines that we have to value social science as a key to success in this process. We have to engage the public, people like you, and talk to you and say, what is this vaccine all about? We have to inform the public about benefits, risks, and supply. And we have to give them expectations. And we have to communicate in meaningful ways, crowding out the misinformation or wrong information. And we have to earn public confidence at a time when earning public confidence is challenging, we all must admit. And we have to earn their confidence that the allocation and distribution of this will be even-handed and fair. We have to make the vaccine available in safe and familiar, convenient places like grocery stores, church parking lots, schools, your doctor's office, people that you trust. It has to be available. And we have to have independent bodies that review the information, review the plans, and instill public ownership where people can have a voice in what's going on. And all of this is very important. So let's talk a little bit about vaccines. There are basically 4 different kinds of big buckets that we are looking at to create a vaccine. One is actually using the virus itself, and investigators in China are using this, a modified kind of virus, to actually what we call attenuate it or make it less dangerous to people. We are attempting to do that kind of vaccine. There's the nucleic acid vaccine, which is the one that we are using with the Mrna trial, where we actually put in a small code into a cell, and the cell then produces the proteins like the virus and allows us to develop an immune response. There are other viruses that are other cold viruses where we put a little piece of the COVID virus inside there, inject that in, and that allows that to reproduce itself. And then we develop a response to that. And then there are small protein segments of the virus itself that we inject in people. And those little small segments can induce an immune response. All of these are meant to get our immune system revved up and the ability to respond to a vaccine. So if we look at the New York Times, I always like to think of this as a scientific journal. They seem to publish things faster than anything else. We see that there are more than 38, I think there's even 40, there's over 142 vaccines being considered, but 38 are in phase 1 as of today, recently, November 10th. 14 are in phase 2. 11 are in phase 3 all around the world, which is the final stages. And some of them have even already been approved on a limited basis in different countries, none in the United States as yet. The whole idea is, can we make antibodies and an immune response to prevent this infection? So let's talk about a couple of these vaccines and we'll leave time for your questions. So I'm going to talk about 2 of them because in the interest of time, and the 2 that have been in the press within the last week. So Pfizer had made a vaccine and they presented some initial data from their study where they looked at 76 individuals and they enrolled these individuals, 45 of whom were enrolled, into 3 different groups. They got this initial vaccine that they partnered with a biotechnology firm from Germany. 12 got the vaccine at 1 dose of 10, 30, or 100 micrograms. And then 3 people were assigned a placebo in each group. And then they studied these individuals, and then they wound up giving them a second dose of the vaccine in the lower groups. They did not do it in the higher group because they found there were too many side effects and that they were getting the response they needed in the lower group. So what did they see with side effects? So here are the side effects that they were seeing with the Pfizer vaccine. It hurt, right? Not a surprise. When you get a vaccine, it hurts. And what they found with the 10 micrograms is more than 50% of people reported some mild pain. When you got the 30 micrograms, it was a little bit more moderate. And when you went to 100, it was even severe. So that's why they really abandoned the 100. People in the placebo group, a third of those people reported pain. So it hurts whenever you get a shot. Very few people reported redness, not very many, and not very many reported swelling. So local effects were mostly centered around pain, and they were either mild or moderate with the Pfizer vaccine. Now this slide's a little bit more complicated, and we have 2 spots. So let's look at the top spot first. This is the first dose. And here we're talking about how did it make you feel? Because when you get a vaccine, sometimes I hear people say, when I got the flu vaccine, I got the flu. What's actually happening is you got the flu vaccine and you got an immune response. That immune response indicates your body is pretty strong and doing exactly what we want it to do. It's responding to the parts of the flu and creating some symptoms that look like the flu. So some people get a fever, not very many. There were under 10% that got fever with this vaccine on the first dose. Sometimes you felt fatigue. Sometimes you had a headache. Sometimes you got chills. That sounds very much like the flu, but that's your immune system working, generating antibodies, generating floating chemicals, sending signals that at the same time it builds an immune response. It doesn't make you always feel terrific. You have a little bit of diarrhea, muscle pain. So they found that these symptoms on the initial dose were more severe in the 100 micrograms, so they abandoned that dose. This is the second vaccine. And what we saw in that was the side effects were more than the first time you got the vaccine. but most were either mild or moderate. Now, what does mild and moderate mean? Mild in our world means that you feel something, but you don't even have to take a pill for it, like a Tylenol or something. It just made you a little uncomfortable, but you got through your day. Moderate means sometimes you take like a little bit of a Tylenol or ibuprofen or something, and that makes it feel better. So in other words, you're doing something to maybe treat that headache, treat the fatigue or the fever, and that made you feel better. Severe means that well, it interfered probably with your day and you couldn't go to work, you couldn't go to school, maybe you had to lay down. And a grade 4 meant you had to go to the hospital. So remember, there's nobody going to the hospital with any of these side effects from the Pfizer vaccine, the second vaccine. Okay, now did it work? Well, this is a complicated graph showing basically, yes, it did produce the immune response we wanted. So this, we're looking at the actual site where the virus normally binds. And all this green is the antibody responses after about 21, 28, or 35 days. When it really revs up, it shows that it's binding that we're generating these antibodies that bind exactly where the virus would normally bind. And that was much better than placebo. Placebo's this gray and it didn't do anything. And this black right here are people who actually had COVID and they looked at their serum, they recovered from it. And so what we're seeing is people who recover get better, that these antibody responses are very similar, if not even higher, than people who were sick with the disease. And when we look here, does it actually neutralize the virus? Does it bind it and stop it? We're seeing that at 28 days, we're getting very good neutralizing effect, even better than what we see with people who actually recovered from the virus. So this is very strong scientific data showing that this vaccine works and is generally well tolerated, not meaning that people don't feel anything, but that the side effects are manageable. So here's the big study that we heard about a week ago, the Pfizer study, where they presented this information publicly, but not all of the information. And this was actually a unique study that they started first with phase 1. They finished phase 1, they halted for a second. If it looked okay, they went to phase 2, which they did with the 2 different doses, the 10 and the 30. They went through that. They stopped for a minute. It looked good. And they went on with the 30 and they went to the phase 3. And they enrolled people now as young as 12 years of age. They've extended it out and they enrolled people throughout the lifetime of their population. And they were seeking to figure out, could they prevent confirmed COVID-19 disease in people who had never had the infection before? And could they be sure that there were not terrible side effects from this vaccine? And they gave people 2 doses of the vaccine in the phase 3 study of the 30 micrograms, 21 days apart. And then they presented this press release just a week or so ago. It was November the 9th. And they said that they started their enrollment July 27th. They gave their 2 vaccines. They enrolled 43,538 people, most of whom have gotten 2 doses, but not all of them. They analyzed 94 cases of COVID disease, and they found it provided 90% protection, meaning that there were only 9 people who got the vaccine who actually had COVID disease. The rest were all in the placebo arm. And so after 2 to 3 months of observation, this is a pretty amazing result. We don't see this with very many other vaccines. This rivals vaccines like polio, measles, mumps, German measles. So vaccines that we know work to protect children from terrible diseases. What about the Mrna vaccine? So I'm not going to present all the preliminary data there. I'm just going to present their data in the interest of time. They're doing a phase 3 study in adults 18 years of older of the vaccine. They're going to see, does it work to prevent COVID disease? And is it safe? And does it cause us to react to these two infections so that we generate an immune response? And they gave a 100 microgram dose of this 28 days apart versus placebo. There were 30,423 people enrolled in this study. 30,423. They looked at their first interim data and reported this on Monday, yesterday. 95 people with confirmed COVID. Only 5 of those individuals were actually in the vaccine group. The rest of them, 90 individuals, were in the placebo group. And this met strong statistical criteria demonstrating almost 95% effectiveness. They also looked at severe cases, who had severe COVID. Those are people mainly who wound up being hospitalized, being sick, could have been on a respirator. All 11 of the severe cases were in the placebo group. There were no severe cases in the group that actually got the real vaccine. Of those 95 people, this included 15 people who were more than 65 years of age and 20 participants from diverse communities, including Hispanic or Latinx communities, 4 Black or African Americans, Asian Americans, and multiracial individuals. So they're just trying to say that their study is diverse and they're looking at diverse individuals. Now, what about the side effects? So the majority of the side effects were mild or moderate. Remember, the grade 3, those are the ones where it might interfere with some activity. And they looked at those where there was more than 2%. And that included pain at the site of injection. It hurt. After the second dose, they noted fatigue in almost 10% of people, muscle aches in almost 9%, joint aches in 5%, headache in 5%, and pain and redness at 4 and 2% at the site of the vaccination. So these more, manageable but interfering effects were present in the study, but they were present in less than 10% of people. And most of these side effects lasted between 2 to 3 days, not much more than 5 or 7 days in most people. So their observation was that it suggests that this has safety and does work, especially evaluated across all the subgroups. There was nobody who this wasn't really working in, and there are enough people in this study from all backgrounds to be able to tell. And that's also true of the Pfizer study. So here in Cincinnati, at the University of Cincinnati, we enrolled 185 people. And you can see that almost 50% of these individuals were from underrepresented backgrounds that normally are underrepresented in studies. So we had a large proportion of individuals, 25% who were Black or African American as self-identified, and 20% or more as Hispanic or Latino, and then more than one race or Asian. So our population was much more diverse than the other study because we spent a lot of time making sure that we tried to recruit individuals to this study to invite them to participate as being representative of our community. Now, what's happening in the pipeline? There are other vaccines that are coming. The AstraZeneca vaccine is being studied and the Janssen vaccine is being studied. And we will be doing the Sanofi vaccine trial that's probably going to start sometime in January, another very large trial, slightly different product from the Mrna or Pfizer vaccine. We'll probably hear results from the AstraZeneca and Janssen vaccines perhaps sometime in December or even early January as well. So there's going to be a lot more information about other vaccines too. And these are vaccines being developed and having support from the US federal government. So to summarize, we have been able to develop rapidly vaccines that produce immunity for COVID-19. And our preliminary data is suggesting in humans they're safe, they elicit good responses, and the initial trials are showing in phase 3 that we can protect people from disease in the early phases of this within the first few months. We do need more data on both safety and effectiveness out to 6 and 12 months. We need more safety data in humans. But I anticipate that by March, we're probably going to see at least 4 vaccines that are likely to be available to people to get themselves. And the vaccine distribution is likely going to start in January here in the United States. And that's going to be based upon the availability of the vaccine and different groups of people. So let me come back to that point for 1 second. There are an estimated 110 million Americans who are either over the age of 60 or have some underlying risk that place them at higher risk for COVID-19. These are individuals that would be targeted first to get some vaccine as opposed to others. And of course, we want to have people available to take care of all of us when we're sick. So the very first individuals that'll probably be receiving vaccine in January will be healthcare providers and first responders, nursing home staff. So right now, Mrna says they have 50 million doses manufactured because remember, they were betting that their vaccine would work. Pfizer also has at least 30 million doses. We're not sure, they may have much more, but they're also a worldwide study. So some of their distribution will go other places besides the United States. So we don't know yet exactly how many doses will be available initially, how many people will be able to get it, but you can see that even if we had 100 million doses, that wouldn't be enough. That's only enough for 50 million people. So this vaccine is going to have to be rolled out in waves to different groups of people, and there'll have to be a fair and equitable and transparent way that everybody knows this is how it's going to go. If you want to learn more about the research studies we're doing, I just put this slide up so that you can learn more about that. And then I really wanted to thank, number one, all of the study participants who have generously agreed to do this study. It's an amazing contribution they've made. In my mind, they are the heroes and heroines of this pandemic because of all of their work. And I really want to thank all my staff. This is our staff on Monday outside the building where we're doing a lot of our research on COVID. And they have been working tirelessly many hours, many hours, trying to help all of us. So I thank you for your attention, and I'm happy to answer questions with Dr. Powell and engage in a spirited discussion.
Dr. Victenbaum, thank you. Dr. Powell, thank you. Now we're at almost 11:30, so hopefully want to be mindful of everyone's time. We did say that this would be an hour, but hopefully you're willing to stay on for about 10 or 15 minutes more. Dr. Powell and Dr. Victenbaum, does that work for you both?
Yes.
Great. So we have a number of questions, and in fairness, I want to start with the first question that was asked, and it was from Robert Brown. And I guess to summarize his question, he's saying that, absent a comprehensive national strategy to mitigate the threat of the COVID virus, how do we persuade people to be diligent about containing this contagion? Meaning, doing the 3 things that we keep reinforcing: wearing a mask, washing your hands, and social distancing. Either of you like to respond?
I'll leave that to Carl, because if I knew, I would really tell you.
So I'll tell you, if I have a conversation with someone and I say, this is what I want you to do, they look at me cross-eyed and they walk away, because we have this notion in our country of this individualism, and I'm going to make my own decision. And so we need a national strategy. We need people to step up. We need all of our scientists to step up who believe this. We need a lot of our respected individuals in society to step up. We need entertainers, sports figures, other people to step up to really say, this is what we need to do. We need to wear a mask. We need to social distance. We need to sanitize our hands. We need to keep our hands away from our eyes and our nose and our mouth. And we have to be very careful about eating and close contact with family, friends, and others during this surge. And, you know, I think we need to get back to community, because what we're seeing is that the individual's approach is not working for us.
Yeah,
Thank you for
I
that.
agree totally. I agree totally with Dr. Victenbaum. And one of the things I would say is the same thing I'll say about clinical trials, and that is to engage and make sure the people that have the trust of the individuals have the right information to share. Because as he said, it is a political issue, unfortunately, and when it should not be. And so the only thing that I can add is to make sure that people who are trusted by the community have the right information to give input to patients and other people that they come in contact with.
Thank you. And thank you, Robert Brown, for that question. So Harold Taylor had a couple of questions, and one of them spoke to the misperceptions about not wearing a mask. I guess Senator Rand Paul said that people who have already had COVID should not wear a mask, and then not worry about the pandemic restrictions. Is this scientifically correct?
No, there's no data or evidence that Senator Paul has any information whatsoever. No one has tested that. That is merely his opinion. And we have no idea how long people shed virus for. Furthermore, we don't test people to see if they are shedding live virus or not. We only test people typically for pieces of the virus or genetic material. And I am unaware of any scientific study published that demonstrates that people who have already had COVID do not need to wear a mask. In fact, many of us are concerned that we may not have lasting immunity. So the last thing you want to do is create a double standard in society. And that's what this does. Everyone should wear a mask, especially Senator Paul.
Can I say something related to that?
Sure.
I apologize. Dr. Victenbaum, has anyone discussed people that have had COVID, whether or not they should take the vaccine?
Yeah, that is a great question, and one that we don't know the answer to. My feeling and thinking is that we probably should vaccinate them, because I see no harm, but I would like to see some safety data around that issue, because I do think that we should have a group of people who've already had it get vaccinated and make sure there is no safety concern. Because sometimes if you already have an immune response, and I can make a scientific argument, we already know after the second dose of our vaccine that the immune response is a little bit more powerful, and that people do feel a little bit more side effects. So what about somebody who's had the full disease and we give it to them? I think we should have some scientific information before we advise people one way or another. I think it's a great question, and we don't know the answer yet.
Okay. Thank you both. Follow up to that question, and I think this might be a general one other comment was about, with all of the rising numbers in COVID cases, should the school shut down in Cincinnati?
Yeah. Well, my personal opinion is where we are right now, we should not be having in-person school, and that we should close that back. I personally would close it back till after the holidays to allow us to calm things down, to organize our plan to be able to administer the initial vaccines in the area. And I think that that would be a useful thing to do.
Okay. Dr. Powell, any response?
No
Okay.
comment on that.
So, from Deborah Wilcox, one of the things that the Health Gap and many of our partners are concerned about is the conversation on mistrust. And obviously, the more informed we are, the more we can begin to break down some of the information and understanding of what we can do and should be doing. Is there any type of implementation of a strategy to build trust around these vaccines in the Black community? Dr. Powell?
Well, of course, Renee, I'm sure you know what I'm going to say, and that is the way we've talked about trust and the way we've dealt with and trying to address that issue for over 20 years is, first of all, getting people involved in the process from the beginning and understanding the process and getting diversity at every level of the process. And what I mean when I say that, the people that design the trials, the people that are in IRBs, the people that investigators, not just patients, people that can influence the design of the trial and can understand the scientific data that leads one to develop the specific vaccine and to conduct the specific design of the clinical trial, and make sure those people are part of the process and can share that information with the community. I showed a picture earlier in my presentation. I showed a picture of with the Belmont report with Dr. Dorothy Height. She was on the commission that wrote the Belmont report. We got to have people like that at FDA, every part of the process, to build trustworthiness in the process. And what my experience has been with Project Impact is the more we have people to understand these processes in information and get the right information from people that they trust in a way that they can understand, that's the way we build trustworthiness. What we're advocating for is more ownership of the entirety of the process, meaning designers, investigators, and not just patients in the clinical trials. We gotta stop being victims and start being the people that really understand and can make these kinds of conduct these kind of clinical trials for our own benefit as well as the benefit of the people around us.
Thank you, Dr. Powell. I'm going to try to wrap up here with a couple of the final questions. Dr. Henderson asked, have the Moderna vaccines developed in the past shown long-term efficacy and safety? And then part two of that, did the Pfizer study analyze the severity of the disease in the placebo group that became infected? And finally, when will study participants who got the placebo get the actual vaccine?
Okay, I think I lost the first part of that question.
So
I got
I didn't know if I read it right. It said Mrna, but I'm assuming it was Moderna vaccines developed. Well, maybe it's vaccines developed in the past shown long-term efficacy and safety. It says mRNA.
Yeah, so the mRNA technology vaccine is not one that has been licensed previously. Personally, as a scientist, I'm very interested in this because it's very scalable. It's easy to make. It's much easier than other vaccines. And I think it will be the wave of the future. So I think this will be the first kinds of vaccines that get approved like this. And I know that that can always make people a little bit nervous because the first of anything is always a little bit nervous. Although being the first on the moon was kind of a cool thing, right? You know, being the first to set the record in some sporting event is kind of a cool thing. So being the first is not always terrible. It can be a little nervous though. So yeah, there aren't the vaccines like that. As far as the placebos go and the data with Pfizer, they presented very limited information so far. It's not been scientifically reviewed. They haven't had the full information, so I really can't answer the questions about that. Moderna presented much more publicly available information, which I was very pleased that they did that. As far as the placebo, I can tell you right now, we are having very hot discussions about how to handle those people getting placebo. The most important thing is we need a little bit more safety information as well as effectiveness information in all of these studies. It's really too soon for us to be giving people the real thing, because we really need to have a little bit more observation period from a scientific perspective. But we are discussing, once the vaccines become available and there are people in the study who might be able to get a vaccine outside of the study, what do we want to do? And as investigators, we're discussing this. We have ideas. We've spoken with Dr. Fauci. We've spoken with the leaders, and we're trying to present these ideas. And we have to get feedback from the FDA on whether these ideas are correct or not. So there will be information coming in soon on how this may be handled. Clearly, we, the investigators, are not going to stand in the way of someone. If there's a vaccine that comes in the community and you're one of the groups that's eligible to get it, we are not going to stop you from getting it. So that's important. And I do want to address one other question on trust. I know people are asking a lot about trust. Trust is something that you build over time, that you work with people, that you listen to people, that you take leadership from other people, that you answer questions, that you show you are committed for the long run, and that you speak a language that people understand and relate to, and they trust you, and that they can test you over time. Trust is not something you build immediately. I'm here in this community. My children went to public schools. I wanted them to have a diverse education. I'm here to fight for social justice on any cause. I am here for the long haul, but I don't expect people to trust me just because they've heard me today for the first time, or even tomorrow. It takes time, and I think you just have to keep at it, and you have to answer people's questions and never say to them, that's a silly question, that's ridiculous. Anything that people ask is important to them. You have to take the time to answer their questions and do it in a way in which it makes sense to them. But we need more leadership from the Black community on all fronts of investigation, vaccine development, science, etc., and to be involved in these decisions. So hopefully, as the Ohio Health Department begins to think about a vaccine strategy, some of our leaders really need to step up, and we need to try and make sure that we really speak to all of these issues, and make sure that the welfare of the Black community is heard and is respected. And I'm really going to have to go. I have
Well,
another.
I wanted to say thank you. We've gone past the 10-minute mark, and I want to thank you, Dr. Victenbaum. I want to thank you, Dr. Powell. What I want to say to all those who are still with us is that we will have a part two. Clearly, we touched the surface with providing information. I think now we need to go deeper into asking and responding to questions and concerns. So hopefully both of you will be willing to come back in the next few weeks, and we'll work with you to do that. And then finally, I want to thank our partners through the COVID-19 communityresources.com site that was launched in March as a place for trusted information for the Black community and the entire community. So visit our site. This broadcast will be placed there. And in addition, I want to thank the African American Chamber of Commerce, the Urban League of Greater Cincinnati, the NAACP, the Cincinnati Medical Association, and Dr. Powell is a member, and they have been a phenomenal partner in all of this work. Councilmember Jan Michelle Kearney, Commissioner Melba Moore of Cincinnati Health Department, and Commissioner Greg Kesterman of the Hamilton County Health Department. We all are in this together, and I am convinced that if we continue to talk, share information, we're going to inform and create greater understanding and knowledge so that there will be greater trust over time. So thank you all for participating, and we look forward to part two. And you all have a great day, and please mask up, social distance, and wash your hands in the holiday season. Thank
Take
you.
care.
All right.
Bye bye, everybody.
Bye.
Thank you, Dr. Powell and Dr.
Thank you.
Victenbaum.